In the second video on membrane potentials, I'm going to talk about the resting membrane potential. After you watch this video, you should be able to define the term and explain how it is established. There's a few terms that we're going to be using more later. You need to define and then be able to use polarization, depolarization, and hyperpolarization. You're going to need to understand this well enough so you can predict how manipulations of the neuron or its environment changes the resting membrane potential. And then finally, be able to name and explain the function of the three types of ion channels I discuss here. As I mentioned before, electrical potential is the amount of charge separated in space. It can be measured in volts or millivolts. Thing I didn't mention before is it's a comparative measure. So what do I mean by that? So if we look and we have positive charges separated in space from negative charges, if you look at it, the number tells me how much charge is separated. So in this case, we've got 10 volts worth of separation. Now remember, the volt is telling me the size of the potential, which is basically how much work it can do when we let these things move. Now with the membrane potential, if you look down here, this would be plus 10 volts, this would be minus 10 volts. So the number tells me how much charge is separated. The sign is going to tell me whether there's an excess of cations or anions here. So the difference between these two numbers is not 20. This isn't a number line. Okay, so we'll talk in a second. What happens if I let some of these move together and intermingle? Then I have less charge separated, right? So if we do that, we'll go from 10 to 9 to 5 to 2. What number do we get at when all these charges intermingle and there's no separation of charge? That's when it'd be 0. So 0 equals no separation of charge. So the difference between these numbers is 10, right? So if we allow this charges to intermingle 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. Okay, so the difference is 10, not 20. All right. So consider the sign separate. It's a separate thing. Okay, it's just the number that tells us how much charge is separated. Now I used that hypothetical example where we've got 10 volts worth of charge separated. When I say it's relative, when it's a comparison, right, this plus or minus, on its own, it doesn't mean anything. I have to compare it with something. Because let's say we've got this many positive charges here. If I've got many, many, many more positive charges here, I can still have a separation of charge. Okay? Compared to, I'm not going to draw them out, but let's say I have 1,000 positive charges here. Now there's a big separation of charge. I have more positive charges here. So again, let's say we set it up so the difference in charge between here and here is 10 volts. In this case, over here, this would be plus 10. This has less, right? So it would be minus 10. Okay. So again, the number tells you how much. The sign tells you whether we have a net excess of positive or negative charges. And we have to compare it with something. So here, in this case, obviously, we're going to compare this space here with this space here. Now, if we let things move down their electrostatic gradient, how are things going to move? Well, these things are attracted to one another, right? So they're going to move together. And let's say we let some of them go, and we get a situation like this. Okay, in this case, we have less charge separated, so that number is going to be smaller. Here, hypothetically, we go from, say, 10 down to 5. Okay, we have less charge separated. This is still positive because we have the excess of positive charges over here compared to this, which would be negative. Now, when we go from 10 to 5, that means less charge is separated. That is going to be called a depolarization. Okay, so first of all, when there's no separation of charge, there's no polarization. The moment we get a separation of charge, 0.01 volts, a very small separation, now we have polarization. Okay, so polarization just means a separation of charge. When we're talking about changes, when we make it a smaller difference, less charge separated, that will be a depolarization. Okay, so again, if I have 10 volts, I have a relatively large degree of polarization. I have a relatively large separation of charge. When that goes to this case here, you can see that we're depolarizing. We're reducing the amount of polarization. The opposite of that is if we would go from this, and then let's say we use pumps or something to grab the charges and separate them. So we're increasing the separation of charge. We call that a hyperpolarization. So if we go from 5, say back to 10, we're increasing the separation of charge, hyperpolarizing. Okay, so the key words here, polarization, depolarization, where we're reducing the separation of charge, and then hyperpolarization, where we're increasing that separation of charge. So here, I haven't drawn anything in terms of a cell. We're just talking about space. Okay, so a potential is a separation of charge in space. I mentioned that there's a bunch of different kinds of potentials that give you information about what's going on with each. So here I want to talk specifically about a membrane potential. Okay, so we're going to add a word. So a potential, you know what that is. Now we're adding membrane. So instead of being separated across space, now the charges are separated across the membrane. Okay, so if I say cell membrane, right, if I talk about a membrane potential, I have to be talking about a cell. If I just say potential, I could be talking about anything. Right? But a membrane potential adds information. Here we're talking about the separation of charge across a cell membrane. Now when we talk about the membrane potential, often we say the membrane potential of the cell. Well, we also are going to have to consider the membrane potential locally. So it's not the same everywhere in the neuron. So if you remember, we have dendrites. We'll have an axon. Sorry, I'm a horrible drawer. All right. But we have the cell body, the soma. We have dendrites. If we look at the membrane potential, and I'm just going to use the abbreviation V sub M. Okay, that's the membrane potential. If I look at V sub M, say here, it might be different than V sub M here. And that might be different than the membrane potential down here. Okay, so these don't have to be the same. This is very important as we move forward. When we're talking about membrane potentials, you have to remember, where am I? Ask yourself that question and be sure you can answer it. Am I in the dendrite? Am I in the axon? Am I at the axon hillock? Okay. Now, sure, the membrane potential up here has a small effect down here. So they do affect one another, but they're not necessarily the same. This is really important for you to remember as we move forward. There's many different types of membrane potentials. As I said, remember, it's a separation of charge across the membrane. When nothing is going on, the neuron's just sitting there. It's the resting membrane potential. That's what I'm going to spend time talking about in the rest of this lecture. Later, we're going to talk about the action potential. This is also known as the nerve impulse. This is what has meaning and transmits information. We'll talk about EPSPs and IPSPs. EPSP stands for excitatory postsynaptic potential. IPSP is inhibitory postsynaptic potential. Okay, so this is just a change in the membrane potential. These occur on the dendrites. The action potential is going to occur along the axon. Okay, so just be aware. The membrane potential is a specific type of potential. There's many different types of potentials. Each gives you more information. Each is more specific than just potential or membrane potential. And that the membrane potential can be different on different parts of the neuron. All right. So before I showed you where we had 10 volts of charge separated, right? That was just a potential. Now, if, let's say this is the extracellular fluid and this is the intracellular fluid, this would be the cell membrane. That phospholipid bilayer that's semi-permeable. If we're in this case where I have a separation of charge across the membrane, now we're talking about a membrane potential. When these things are measured, again, we're going to use some equipment, but we're always going to compare inside to outside. Okay, so we're going to learn that the resting membrane potential is minus 70 millivolts. Okay, so if I just tell you the potential is minus 70 millivolts, what do you know? Well, you know that there's 70 millivolts of charge is separated. Where's the positive and where there's negative charges, though? That you don't know unless I give you more information. Okay, so here, if we are always comparing inside to outside, and you look at this number, what that tells you is that the inside is slightly more negative than the outside. Okay, so if I tell you the resting membrane potential is minus 70 millivolts, what you know is the inside is slightly more negative, and this is how much charge is separated. Okay, this is a little bit better diagram showing how most of this is measured. If you think about membrane potentials, action potentials, and things like that, you have to have access to one neuron and one axon. In humans, that's pretty hard. How am I going to get a single neuron? I can access your brain, I guess, but I have to put you all back together at the end, right? It's also incredibly small. It's very difficult to get a recording electrode into one neuron in a living, awake animal. There's an exception. So this is a squid, sotong. I like sambal sotong. So here we've got our squid, our sotong, and within it, there's something called the squid giant axon. So if you look for it, you can actually see a single neuron with an axon that's 1 millimeter in diameter. That is huge. It is just one cell. It's not a nerve. Nerves are bundles of thousands or tens of thousands of axons. This is just one axon that's 1 millimeter in diameter. This thing is huge. This is great for neuroscientists because it's really easy to access and it's really easy to record. Okay? So what they do for a lot of experiments, a lot of the stuff we know about the action potential that I'm going to talk about in the next lecture is done in this squid axon. You're going to have two electrodes. You'll have a reference electrode and a recording electrode. This recording electrode is going to go into the squid giant axon. And essentially, we're going to use a voltmeter to measure the difference between the reference electrode and the recording electrode. Okay, so in this case, when we first start, if I've got the reference electrode and the recording electrode in the saline solution, here they call it seawater. I mean, we don't call it that in the lab. It's saline, a salt solution. If you look, these are in the same fluid, right? There should be no separation of charge in the seawater. It should be the same relative concentration of cations and anions throughout. So if we look at these, there should be no difference between charges inside, between these two electrodes. And so if we were to look, our polarization, our voltmeter should read 0. There should be no separation of charge. Now, if we look down here, then we take our recording electrode and we put it into the squid giant axon. You know that the inside is slightly negative compared to the outside. So now, inside compared to outside, there's about 70 millivolts worth of charge separated. That means the inside is a little more negative, so minus 70 millivolts. That's what we're going to see. Okay. Remember, inside to outside. For this next question, I want you to don't get confused. So this isn't how we normally talk about it. But what if I wanted to know, for some reason I wanted to talk about it for the outside compared to the inside? Let's reverse that. What's the membrane potential outside compared to in? Well, you know it's 70 millivolts. That's how much charge is separated. But the outside has more positive charges compared to the inside. Okay, so then the outside compared to the in would be plus 70 millivolts. Okay, but by convention, we always compare inside to outside. All right. So, as I mentioned, this is so important. I'm going to say it over and over. Inside compared to outside. The number tells you how much charge is separated, and the sign tells you whether there's more anions or more cations on the inside. If it's a negative charge, that tells you that there's more, a negative number, that tells you there's more negative charges on the inside. If it's a positive number, that tells you there's more positive charges on the inside. Okay, so for example, if it's minus 10 millivolts, if V sub M, the membrane potential, equals minus 10 millivolts, you know that there's a separation of charge, 10 millivolts worth, and the inside is negative compared to the outside. What if I told you then Vm is plus 10 millivolts? Well, in this case, it's the same separation of charge, but in this case, the inside would be slightly positive, so there'd be an excess of positive charges inside the cell. What if the membrane potential was 0 millivolts? What does that mean? The number, again, tells you how much charge is separated. 0. No charge is separated. So here, in that case, the amount of positive and negative charges inside and outside the cell is the same. I want you to remember minus 70 millivolts. This will be the resting potential, the resting membrane potential. Right? I haven't explained everything yet, what rest means, but I will in a second. Just remember that number, minus 70 millivolts. All right, so here I have a simple diagram. When I say the neuron's at rest, nothing is going on. There's no stimulation, no changes, nothing. It's just sitting there. Now, this is not very realistic. This never, ever, ever, ever happens in real life. Each cell, you may recall, has about 10,000 connections, 10,000 things trying to change the membrane potential. One of those is always active at some time. Okay, so the neuron at rest is actually a fairly rare state. All right, that being said, we're going to start here. We've got salt water, sodium chloride outside. We got potassium inside. We have these big organic anions inside. So when we look at all of these things, there's going to be a slight excess of negative charges on the inside. Okay, not a lot, but a slight negative net charge on the inside. Minus 70 millivolts. Now, if you look, we talked about this before, the electrostatic forces, where like charges repel, positive charges are going to attract one another. And the force of diffusion, where molecules want to move down their concentration gradient. And we've been through this already, but just real quick review. Sodium, the force of diffusion is driving it in. The inside has all these organic anions, so it's negative. The positive charge is going to be attracted to the inside. So for sodium, both the force of diffusion and the electrostatic force are trying to drive it into the cell. Now, quick question. Why doesn't sodium just move into the cell? And the answer for that is charged molecules, ions, can't pass through that membrane. And in the situation where the neuron's at rest, there are no ion channels open for sodium. So sodium wants to go in, it just can't. For chloride ion, higher concentration outside, so the force of diffusion is driving it in. It's negatively charged in here, so negative charges will repel. So the electrostatic force is actually going to repel, push chloride out of the cell. Potassium, higher concentration inside, so the force of diffusion is going to try to get it to go down its concentration gradient from inside to out. The electrostatic force will try to bring it back in. Now, as I mentioned before, if a channel does open and the molecules, these ions, can move, it's the force of diffusion that's stronger. Okay, so if I'm asking you what would happen if I changed the flow of this or that, in our case, in the situations where I'll ask about, we can ignore these electrostatic forces. Pay attention to the force of diffusion. All right. So at rest, there are actually special leak channels. These are for potassium only. So if our neuron is sitting there, again, sodium chloride outside, potassium inside. If I have a few leak channels, these are going to allow some potassium to flow. So which way does potassium flow? Again, down its concentration would drive it out. The electrostatic force would try to bring it back in, but I already told you the force of diffusion is stronger. So potassium is going to leak out slowly. Now, what effect would that have on the membrane potential? We'll go through this in more detail later, but if this goes out, I'm losing positive charges. That's going to make the inside more negative. So fewer positive charges equals more negative. That's going to hyperpolarize my cell. It's going to change the membrane potential. When we get to the action potential, you'll see the effect that will have on the cell in the long run, it turns out that's bad. That's very bad. So we need to counteract that efflux. Efflux means flow out. So we have to counter the efflux of potassium. Okay. So at rest, sodium and chloride are not moving. Potassium is slowly leaking out through these leak channels. In order to stay alive, we have to counteract that. And the way we do that is something called the sodium-potassium pump. So the sodium-potassium pump, you can see, is a big protein that goes through the membrane. It's essentially going to exchange sodium and potassium. It's going to take sodium from inside the cell and pump it out. Okay, so in terms of sodium, it's going to help maintain a low concentration inside by getting rid of that sodium. It's then going to take potassium from the outside and bring that into the cell. Okay, so remember, some potassium was escaping through those leak channels. The pump is going to counteract that. It's going to grab some of that and bring it back in. Now, the thing about the pump is it requires energy. Okay, so ATP is the currency of energy. So for every ATP molecule, 3 sodiums go out, 2 potassiums come in. So the function of the pump is to counter the leak of potassium. Okay, so that's one thing I want you to remember. The second thing is it's going to help maintain the concentration gradient. So if the pump stops working, you're going to have problems with this and this. We'll talk about the implications of that later. All right, so at rest, when nothing is going on, that's what the resting membrane potential is, right? It's the separation of charge in a neuron when nothing is going on. It's established in part by the cell membrane, and then it is a function of those big organic anions and the fact that sodium, potassium, and chloride can't easily flow through that membrane. Polarization, depolarization, and hyperpolarization are words that we're going to use a lot in the next video, so make sure you have that down here. Now, in terms of being able to predict how manipulations of the neuron or its environment affect the membrane potential, if you really understand this, you should be able to predict what would happen if I increase the concentration of sodium in the extracellular fluid. And we'll go through and do some problems where you have to answer those questions so you can check yourself. But think about that. See if you can answer it. If you can't, you missed something and probably should go back and look either in the book or watch the video again. And then the 3 types of ion channels I talked about. With the 3 types of ion channels here, we talked about leak channels, right? That are always open, and I alluded to a couple others that we're going to talk more about. So in terms of the 3 different types of ion channels, we will cover the rest of that in the next lecture.